Hi, this is Jessica. I'm Masters in Nursing. I have been closely associated with my assignment services since past 5 years and have guiding and consulting university students with their various academic projects. Today, I will be taking you through one of the common types of project students are generally asked to compile during their academic coursework. I will guide you through the process of conducting research and compiling high-ended solution with the help of a sample. Let us have a look how to interpret instructions and include them while drafting the final research work. So, the sample today we are going to discuss is about bioinformatics. Bioinformatics is an emerging field that is incorporating various domains, that is computer science, chemistry, biochemistry, biology, statistics, maths, engineering. So, it is the wholesome of all these things that require an structural as well as analysis of a structure that will help us to predict that what will be the changes or mutation that has been taking place in an individual, or how it is going to impact further. Moving ahead, so this is the assessment on which we are going to perform our bioinformatics analysis. So the assessment is asking to choose a gene and its protein product of interest where the genes has been given, that is HNF1A or KCNQ1, that will be selected as the gene, and we have to define that what protein has been expected from this. So the question is asking what gene you have selected, detailing about the chromosomal location, expression patterns, transcription, translation, post-translational processing, information on general function of protein within the cell tissues under normal condition. From this location, chromosomal locations, to post-translational processing, these are the cellular processes that a gene has undergone to make up functional protein. And after that it is asking that what is gene or the protein product is expected to function as. After that, it says bioinformatics that we have to carry out our bioinformatic analysis on selected gene, that is one selected gene, and using a particular software which is UniProt software. UniProt software is a bioinformatics software that is present online that can be accessed with the help of this link. It is going to present the pictorial information of a protein, its motifs, sequence functioning, catalytic site, and where the mutations has been taking place. So, as the question demanding, we have to present the 4 images, that is A, B, C, D, that will help us to understand that what is the gene about, what it is protein family, its domains, what is the catalytic site, and what are the motifs that is present in this particular gene. And we have to present in the form of screenshot. After that, it will ask that if we see that any mutation is there, what we think, that what functional changes or modification we are going to expect if the changes has been take place, and what is the contribution is seen in terms of functionality. So HNF1 is the gene that is related to it is the gene that is located in the hepatocytes, and KNCQ1 is the gene that is presented in the iron potassium channels. So we have to provide an experiment where we have to predict one of the gene is impacting the type 2 diabetes, because both involved in the process of type 2 diabetes and its metabolism. After moving ahead with this, the word count has also mentioned here, so we have to take care this point that the 250 words will be devoted to part 1, 500 words will be devoted to page 2, and page 3 will be comprising of 250 words. Moving ahead with this. Okay, so the page 3, that is of experiment, we have to present the 4C problem in the form of an experiment, that is a flowchart diagram that is describing that how it is impacting and what are the outcomes that has been observed if the things take place in that manner. Now last thing, that is referencing. We have to take care that the GCU library guidelines has been followed while making the Harvard referencing, that is the demand. So it will varies as per the university. and we have to take care that the referencing format will be there, as it will help us to gain marks or lose marks sometime. So defining the part A, we have selected the KCNQ1 gene. That gene belongs to the potassium channel, and we have defined the chromosomal location that is defined here. What is the expression of that gene, what is the translation as it is defined in the previous one, and what sort of function it is performing. Now part B, that is UniProt 1, in which we have to present the protein structure, protein motifs, functional units and structural one. So we have presented the protein motifs, that it is a 4-chain protein, and what are the conserved domains and what is functional units present in this. After that, the further is showing the domains, motifs, and then the regions where the mutations has been expected that will lead to the change in the catalytic site. This is the last one, that is defining the similar protein family in which it is present or showing the highest of similarities. Then comes the experimental part, that is page 3, where we have defined that how KCNQ1 gene is related to type 2 diabetes, and what is the impact will be seen if the increase in glucose is there or increase in insulin is there. That will lead to the change in the functionality of. Thank you. I hope this sample was effective for you and will help you and guide you in your future research project. In case of any query, please reach back to us, as we would glad to assist you in your academic course and research work. Thank you.